I'm going to discuss the invasion mechanisms of complement cascade by, other microbes. Many different types of organisms have developed ways to invade the complement cascade. It's very detrimental to important pathogens. Very detrimental. Um, they can do this by a couple of ways. They can do this by a lot of things. And specifically, I'm going to discuss the Staphylococcus aureus. I'll get to that in a second. But they can do this by, they can invade the complement cascade by producing a thick peptidoglycan layer to kind of like brush off some of the activity of the complement cascade proteins, and this is a type of gram-positive bacteria does this. Their thick cell walls and capsules brush off the MAC complexes, and the rest kind of just hide, you can say, in a way. They kind of just hide in the intracellular vacuoles to escape detection. So they hide so that they won't be detected. Gram negative organisms, they have a shield of like magnesium ions in the outer membrane to help repel some of the individual complement proteins. They also do this by forming a thick capsule, which means that activation will occur, on the outside of the slime coat, which complement cascade never gets close enough to the cells because they're constantly shedding these coats. So that's another way of protecting the invasion. So Staphylococcus aureus, I always get that jumbled up. Staphylococcus aureus is a major human pathogen. It's encapsulated, and it is also gram positive. It's a gram positive bacterial strand. It has different ways to invade destruction by the complement system. These methods inhibit both the classical and the alternative pathways of complement activation. I tried to get, the book has a really, really good detailed picture of this and the different steps. I tried to tried to find it online and I couldn't find it. So I'm sorry, you're gonna kind of have to just go with me as I describe these here without visually showing it to you. But they basically have like 5 different steps, you can say, that they go through on this Staphylococcus aureus. The capsule, which is on the outside, is on the very outside. It's a polysaccharide, and it has the inhibition of opsonation. Opsonation is when a pathogen is marked for ingestion and eliminated or killed by a phagocyte. So they have the inhibition to eliminate or kill a phagocyte. Underneath the capsule, the complement factors can assemble on the cell wall. So underneath the capsule is the cell wall, and complement factors can assemble on this. but most of them do not have access to the receptors. So most of them can assemble because they don't have access to the receptors. And the picture would have really helped me sort of explain this a little better. But it's a enzyme on there called EFB. EFB binds to the C3 region. It prevents it from reaching the cell surface and inhibiting further activation of the complement cascade. So it binds to the C3, and then it prevents it from reaching the cell surface, and it inhibits further activation of the complement cascade. And then next it goes to a protein named SPA. That's a capital S and a capital A. This protein binds to the IgG, and in a way it does not permit that the FC receptor binding. So the protein SPA binds to IgG, and in a way that does not permit FC receptor binding. And from the SPA and the IgG, it goes to another protein called SAK, and that's a capital S, capital K. This activates the plasminogen. Sorry, I probably said that wrong and messed that all up. The plasminogen is a protease, and it's capable of inactivating IgG and C3b. So it can inactivate IgG and C3b. From that, it goes to the clumping factor, clumping factor A, which is abbreviated as CFA. It binds to factor 1, and it localizes in the microbial surface, where it then inactivates any C3b that tries to bind. So once it reaches the clumping factor, it can inactivate any C3b that tries to bind. And I think this is a really good detailed version, and also a really good example of something everybody know of for the complement cascade.